For the first standard of respiration, I'm going to be talking about oxidation. Oxidation is a reaction in which the atoms of an element lose electrons and the valence of the element increases. Oxidation and reduction refer to a change in oxidation number. The actual transfer of electrons may not occur. Oxidation is better defined as an increase in oxidation number, and reduction as a decrease in oxidation number. The breakdown of organic compounds is carried out by a series of redox reactions, where the transfer of electrons determines the oxidation, removal of electrons, or reduction, addition of electrons of a compound. For the second standard of respiration, I'm going to be talking about glycolysis. Glycolysis is a metabolic pathway that is found in the cytosol of cells in all living organisms. The process converts one molecule of glucose into 2 molecules of pyruvate, making energy in the form of 2 net molecules of ATP. 4 molecules of ATP per glucose are actually produced, however 2 are consumed for the preparatory phase. The first phase is the energy expending phase. 2 ATP are used for every glucose molecule that enters into glycolysis. The second phase is the energy generating phase. 4 ATP are produced by substrate level phosphorylation, and the coenzyme NAD+ is reduced to NADH, while the carbons from the glucose are oxidized. For the third standard of respiration, I'm going to be talking about the Krebs cycle. The Krebs cycle is a series of enzymatic reactions that occurs in all aerobic organisms. It involves the oxidative metabolism of acetyl units and serves as the main source of cellular energy. In the citric acid cycle, a single pyruvate molecule creates a single acetyl CoA to enter the citric acid cycle. However, since each glucose molecule produces 2 pyruvates, the actual production per molecule of glucose is twice that shown here. The total output of the citric acid cycle per pyruvate molecule is 2 carbons, 3 NADH plus 3 hydrogens, 1 ATP, and 1 FADH2. Most of the energy made available by the oxidative steps of the citric acid cycle is transferred to the electron acceptor NADH+ to form NADH. For the fourth standard of respiration, I'm going to be talking about oxidative phosphorylation. During oxidative phosphorylation, electrons are transferred from electron donors to electron acceptors such as oxygen in redox reactions. Oxidative phosphorylation utilizes the energy released by the oxidation of nutrients, in this specific case glucose, to produce ATP molecules. The redox reactions release energy, which is used to form ATP. These linked sets of proteins are called electron transport chains. The energy released by electrons flowing through this electron transport chain is used to transport protons across the inner mitochondrial membrane in a process called osmosis. For the fifth standard of respiration, I'm going to be talking about fermentation. Fermentation is the process of sustained substrate level phosphorylation of glycolysis. It does not use cellular respiration or any ETC, therefore it does not require oxygen to generate ATP. Fermentation does require a sufficient supply of NAD+ to accept electrons to sustain the process. There are 2 types of fermentation: alcohol fermentation and lactic acid fermentation. Both involve glycolysis and reactions that regenerate NAD+ from NADH, differing the end products. Alcohol fermentation is a 2-step process. In step 1, CO2 is released from the pyruvate, and pyruvate is converted into acetaldehyde a 2-carbon compound. In step 2, cetahaldehide is reduced to enthol by the NADH. The NAD+ is generated used to sustain glycolysis. Lactic acid fermentation is a 1-step reaction. The pyruvate is reduced directly by the NADH without the release of CO2 to form lactic acid. Lactic acid fermentation uses the following pathway: glucose to pyruvate to lactate. For the 6th standard of respiration, I will be talking about metabolic pathways. Each metabolic pathway consists of a series of biochemical reactions that are connected by the products of one reaction or the substrates for subsequent reactions. Metabolic pathways are often considered to flow in one direction. Some metabolic pathways flow in a cycle, wherein each component of the cycle is a substrate for the subsequent reaction in the cycle, such as the Krebs cycle. Several distinct but linked metabolic pathways are used by cells to transfer the energy released by breakdown of fuel molecules into ATP and other small molecules used for energy.